So our project is blue-fluorescent SN38 silyl ethers, the synthesis, biological evaluation, and membrane trafficking kinetics for topoisomerase-based anti-cancer activity. Next slide, please. Next slide. Okay. Oh, go back one. Okay. So just a brief outline to our project, starting off with just an introduction to topoisomerase inhibition, moving on to the general role of silicon in the current drug discovery space in like past precedents. Moving on to then our own synthesis of C10 silyl ether S30 analogs and their subsequent biological evaluation through like mechanistic conformation, cell uptake, and general potency. And lastly, we'll be talking about some docking. Okay, next. So topoisomerase inhibition is a powerful strategy for anti-cancer therapeutics due to the central role that topoisomerase 1 plays in cell proliferation. So during the S phase of the cell cycle, as DNA unwinds, topoisomerase 1 relieves the generated supercoiling strain by basically nicking the DNA, thereby allowing it to fully unwind. So topoisomerase inhibitors, therefore, sorry, I talk really fast, my bad. So topoisomerase inhibitors, therefore, bind to that topo DNA complex and stabilize it, so that basically doesn't allow the DNA to religate, which then causes permanent damage to the DNA. So when the cell recognizes this, it basically goes into programmed cell death, which is also known as apoptosis. So topoisomerase inhibitors, including our own parent compound, camptothecin, which we'll talk a little bit about later, they tend to share a couple of general features, such as being flat, cationic, rigid structures that tend to have a mix of both hydrophobic and hydrophilic moieties. So some previous FDA-approved drugs that utilize this strategy include some that are derived from our own parent compound, camptothecin, such as irinotecan and topotecan. Okay, next.
Okay. In terms of using silicon as in terms of using silicon in medicinal chemistry, it's actually relatively unexplored. So a really interesting stat we use is that silicon makes up over 28% of the Earth's crust, but it's actually found in less than 1% of preclinical candidate drugs. And what's interesting is that silicon composes of various similarities to carbon in terms of its electronic properties. So it's actually been proven in past studies that attaching these cell ether groups can improve bioavailability and stability. And since our lab loves to work with different natural products, I wanted to analyze some of the past studies that use similar natural products, such as one conducted by a member of our own lab, Tiffany Gu, and other members as well, using the natural product angiographolide. And they essentially attach different siloxy groups onto that natural compound. And what was interesting is that it changed both the biological properties and anti-cancer activity. And what we noticed in that study is that one compound, especially diphenylmethylsilyl, the analog, actually went from over 100 micromolar to around 2.2 micromolar IC50 after attaching that cell ether group onto angiographolide. So that was really interesting. And then two other natural products we wanted to analyze are mycophenolic acid and methoferrin A. And by attaching these siloxy groups, they essentially both help the metabolic stability and the anti-cancer properties. Next slide. Okay, so from this, we coined the term siloxytecans for our analogs, which essentially compose of SN38, which is a camptothecin derivative, and camptothecin comes from the natural product a Chinese plant, Camptotheca acuminata, and essentially we're attaching different cell ether groups to that C10 position. So that's how the name siloxytecans was coined. Next slide. Okay, so yeah, as I mentioned earlier, we're working with camptothecin, our parent compound, and SN38, which is a derivative. And actually attaching different silane groups has two camptothecin derivatives has been studied before. Next slide. Okay, so two past examples include DB67 and kinetecin, which have tert-butyl dimethyl silane and silylethyl groups at the C7 position, respectively. And they've been shown to increase lipophilicity almost like 25 to 50 times more than camptothecin, and also have been proven to reduce both ovarian and lung cancer when tested in vitro, in vivo, in preclinical studies. Next slide.
So as Ashley mentioned earlier, inspired by this, we basically designed and synthesized our own siloxytecans by adding 4 different siloxy groups to the C10 position, which is that hydroxyl, though, yeah, it's highlighted blue. Namely, tert-butyl dimethylsilyl SN38, tert-butyl diphenylsilyl SN38, triisopropyl SN38, and dimethylthexylsilyl SN38. So each of these vary in a slightly different way, and we wanted to test how that would affect their biological potency compared to their parent compound. Next slide. Okay, so the first step in doing so was determining their C log P and TPSA values. So C log P stands for calculated log P and is basically a measure of the lipophilicity of a compound. So the higher the number, the more lipophilic our compound is, thereby meaning that in theory it should be able to permeate cell membranes better, which should in theory increase potency. Next slide. Okay. To start off with measuring our potency, we started off with an antiproliferative assay, such as an MTT assay, by which we were able to determine our IC50s of our compounds. So IC50 is basically the measure of the concentration at which a drug reaches 50% of its total inhibitory potential. In layman terms, like 50% of the max a drug can work. So what we noticed is that across 8 different cell lines, including colon cancer, breast cancer, ovarian cancer, and one non-small cell lung cancer cell line, our compounds display double-digit micromolar to single-digit nanomolar IC50s, which is incredibly potent. In all of our cell lines, they either show comparable or in some cases greater potency when compared to their parent compounds. But what we did notice is that in specifically Calu-1 cells, which are the only non-small cell lung cancer cell line that we tried, both the parent compounds and our siloxytecans show decreased potency, as you can see on our IC50 graphs. We hypothesize that this is due to the membrane composition of those cells. Next slide.
Okay, and what's really interesting about these siloxytecans is that they fluoresce blue under UV light. So here you can see the 4 different fluorescence spectra of our siloxytecans, and each graph shows the excitation and the emission spectra. And the difference between these two excitation and emission maxima is known as the Stokes shift. And for our compounds, that's around like 45. And this is really important because it tells us how much energy is lost between the absorption and emission. And typically, a larger Stokes shift means the spectral overlap is less. So this improves imaging clarity and also reduces like background interference when you're imaging these compounds. And so exploiting these unique blue fluorescent properties, we deduce that we can use this aspect of the siloxytecans to measure them in vitro in live cell imaging. So the PosiLab members will talk about that.
Well, yeah, because they're blue fluorescence or they're blue fluorescent, it makes sense to just go ahead and drug them and check them under a microscope over time course to see what happens over the course of time, right? And we did that by incubating the cells for with a certain drugging time course, and then harvested or rinsing off the excess background compound, leaving just the cells in there. We wash out the supernatant, and then we run it through a fluorescent microscope, and then we analyze the brightness of the cells with image A, and we analyze the mean fluorescence intensity, which is like basically the average intensity of the blue in the cytosol of each cell. And what we did is we actually plotted these over 4 hours, but we cut it off to 30 minutes in these figures because we noticed that cell uptake kinetics plateau from 10 to 30 minutes. And when you put it in a spectroscopy graph or plot like here, you can see that these compounds get into cells like HCT106s and HEP2s relatively well, but HEP293s and CALU1s do not perform as well. So it's helpful to put these in like visual perspectives. And over here we have heat maps for the first 30 minutes. And as you can see, again, cellular uptake of the compound begin to plateau around 10 minutes for all the compounds. For some conditions, intracellular fluorescence peaks before 10 minutes, but that's not the general trend that we see.
Okay, and these are going to be representative images of selective cells from each cell line at both 10 minutes and 30 minutes. And the outline panel highlights the cells drugged by DMT. So this compound consistently demonstrates high levels of intracellular fluorescence across all cell lines, with the exception of Calu-1s. Next slide. We can note that Calu-1s remains comparatively dimmer even when drugged with brighter compounds such as DMT, which suggests that there's difficulty getting into Calu-1s. Next slide. And so here are the mean fluorescent intensity graphs over 4 hours. We can note that Calu-1 remained at the bottom of most of the graphs, and the cell lines had a consistent trend of peaking at around 10 minutes. Next slide.
Okay, so what we want to do next was kind of a mechanistic confirmation of our siloxytecans. We want to make sure that the way that they work is the same way that our parent compounds work, SN38 and camptothecin. So we started off doing this through two types of flow cytometry, apoptosis and cell cycle. So the purpose of apoptosis flow cytometry is to determine whether our compounds also induce apoptosis, just like the parent compounds do. So we did this by staining our cells with two different stains, the green phytzianexin, which stains phosphatidylserine in early apoptotic and late apoptotic cells, and propydium iodide, which is a red stain, which stains DNA in dead cells, so necrotic and late apoptotic. So by doing so, and then by running it through the flow cytometer, we can quantify how many cells are either only green, which would be only phytzianexin, only red, which is only propydium iodide, or both green and red, which would be late apoptotic and have both the stains. And thereby we can determine like the percentage of our cells that are either necrotic, late apoptotic, or early apoptotic. So as you can see from our graph, our compounds do also induce apoptosis, just like our parent compounds do. Next slide. Okay. Our next method of mechanistic confirmation was a more specific flow cytometry assay, which is a cell cycle assay. So SN38 is an S-phase inhibitor, which causes G2 arrest in the cell cycle. So what we want to determine was that our compounds also do the same. So this assay only uses propydium iodide, and by only staining it with propydium iodide, we can determine what stage of the cell cycle the cells are at. So as you can see in our graph, our compounds do also cause G2 arrest, just like SN38 and camptothecin do. And in some cases, especially with dimethylethexyl SN38, which has shown in previous assays as well to be kind of our standout compound, it causes greater levels of G2 arrest than even our parent compounds do. Okay, next.
Okay, and then the last method of confirming the mechanism of action of these compounds was through a topoisomerase 1 assay. And this was gel-based, which means we ran it through gel electrophoresis to determine the results and then quantified it with ImageJ. And so what we did through this assay is we incubated supercoiled DNA, topoisomerase 1, and then our siloxytecans. And what we expect to happen if the topoisomerase 1 inhibitors are functional is you would see an increased amount of nicked DNA, which happens because the topoisomerase inhibitors prevent the DNA from religating. And so as you can see by the gel, we actually saw differing amounts of nicked DNA in the various siloxytecans. And then we compare this against the no topo control, which had only supercoiled DNA, and then our marker. And so using the bar graph on the right, you can see all our siloxytecans exhibit different rates of this inhibition and also different rates of stabilizing that cleavage complex. And DMT-SN38 was particularly particularly good at inhibiting topoisomerase 1, which is consistent with our MTT data. Next slide. Okay, and then lastly, we evaluated the binding affinities of our siloxytecans to the topoisomerase one DNA interface. And we did this by first docking camptothecin to the topoisomerase DNA complex, and then removing camptothecin and then redocking our siloxytecans and SN38. And as you can see by the docking scores on the screen, they displayed relatively similar binding affinities within like -10 to -9 kcals per mole. And also the A to E ring structures are relatively similar in location throughout these binding poses. But what we noticed was that in TBD-PS SN38 actually formed a new pi-cat ion interaction with lysine 425. But other than that, this shows that the steric bulk displayed by these cell ethers doesn't really play a huge role in their binding affinities. Next slide.
So in conclusion, our four siloxytecans exhibit comparable antiproliferative effects compared to their parent compounds. Additionally, they have remarkable blue fluorescent properties, which then allows for real-time tracking of our compound in and out of cells. Additionally, the data that we acquired through this real-time tracking is consistent with our data from the antiproliferative assays. Okay, next.
Okay, and this SN38 study actually connects to another one of our projects, which is the bifunctional project, in which we are conjugating different estrogen receptor ligands onto SN38 to form bifunctional ribtacs. And so as a brief overview of this presentation, we're first going to introduce modern anti-cancer drug delivery strategies and then introduce what ribtacs are and how they play a role in our project. And then we're going to introduce hormone-dependent breast cancer, specifically ER-positive breast cancer, and talk about our synthetic routes, which compose of tamoxifen SN38 and our new teremethine SN38 conjugate, and then we'll end with acknowledgements.
Okay, next. Okay. So modern anti-cancer drug delivery strategies can be grouped into two broad categories: precision drugs and non-precision drugs. So precision drugs are drugs that are able to target a specific type of cell, such as a specific type of cancer cell or a specific marker on these types of cells, for example. Non-precision drugs have extremely high potency, but not necessarily the same selectivity that precision drugs do. So some examples of precision drugs in the drug delivery space are examples such as trastuzumab deruxtecan, which is an antibody drug conjugate, and ARV-471, which is a PROTAC. So these are two different methods of creating precision drugs. Next. Okay, our work focuses on creating ribtacs, which are regulated induced proximity targeting chimeras. What this means is that our drugs consist of an inhibitor, which in our case is SN38, same one as last project, and a ligand, which in our case is going to bind to an estrogen receptor, our target protein. Our effector protein in this case is going to be topoisomerase, as that is what SN38, our inhibitor, binds to, and that is how we kill the cells. So this kind of dual function molecule creates a very, very potent, very selective molecule that is able to selectively target, in our case, breast cancer. Next. Okay, some examples of this type of drug in the past are artemisinin estrogen and artemisinin tamoxifen, both of which are also breast cancer selective. Next. Okay. So the reason that this is a very important or very powerful strategy that we could take is 70% of breast estrogen receptor positive, meaning that they overexpress estrogen receptors when compared to regular cells, both just regular breast tissue and overall, like in the rest of the body. And so by targeting the estrogen receptors that are overexpressed on these cells, we can create our drug that will be able to selectively target breast cancer without harming other cells as much, which would reduce other off-target cytotoxicity. Next.
Okay, so the specific estrogen receptor ligands we're working with are tamoxifen and teremethine, which are both selective estrogen receptor modulators. And these two are pretty similar in structure. They both share the three large aromatic rings, except teremethine has an extra like propyl group at the top, while tamoxifen has an ethyl group. And then we're linking these to the protein inhibitors, SN38, which of course connects to our previous study. And this is a topoisomerase inhibitor. Next slide. Okay, so we initially started with two different syntheses of tamoxifen SN38 conjugates because we weren't sure how attaching the linker onto tamoxifen would affect the binding affinity of it to SN38, which we can later confirm through docking. And so we decided to have two different pathways of connecting tamoxifen to SN38, one from attaching it to the oxygen and one to the nitrogen. And so, and so in this O-link tamoxifen synthesis, we first start with 4,4-dihydroxybenzophenone and do a McMurray coupling to form the olefin between our starting material and propiophenone. And then we do an alkylation with 2-dimethyl aminoethyl chloride. And this is a nucleophilic substitution to attach it to that side chain. And so this opens up the other hydroxyl for us to attach the linker. And these two so far are the only reactions we've gotten yields for. But the next step after that is to do the Mitsunobu reaction to couple our compound with that protected carboxylic acid linker. And then we then cleave the tert-butyl ester using TFAD protection and DCM. And this unmasks a free carboxylic acid, which we later use to couple to TBS SN38 using EDC coupling. And then we remove the TBS group under mild acidic conditions for the final product. Next slide. Okay, and then we follow a similar pathway for N-linked, where instead of 4,4-dihydroxy benzophenone, we start with 4-hydroxybenzophenone, and then that phenol undergoes SN2 alkylation using 1-bromo-2-chloroethane, and then we again conduct the McMurray coupling using propiophenone to form that third compound, and with that, we perform a nucleophilic substitution reaction using methylamine and ethanol under reflux conditions with catalytic sodium iodide. And this introduces that free amine group so that we can attach the linker onto that position. And so we do succinate anhydride ring opening chemistry to introduce that carboxylic acid linker. And again, we attach TBS SN38 using EDC coupling and then remove it using HCl. And next slide. Okay, so these are two tamoxifen SN38 conjugates, but because those two were relatively difficult in terms of both length and also the McMurray reaction is pretty difficult in terms of yield, or like it didn't achieve very high yield. And so we decided to propose a new synthesis using teremethine. And so this synthesis is relatively more simple, composing of two different steps. And it starts with teremethine citrate, which is teremethine and a different acid. And we use an SN2 reaction to using sodium azide and DMSO in order to replace that chloride-leaving group with an azide group. And so initially we used DMF, but then we decided to switch to another aprotic solvent, and this ended up working through test reaction, but we're still working on optimizing this reaction on a larger scale. And after we complete this first step, we want to use an alkene linked to a drug, particularly SN38 or a different drug. And using a late-stage click chemistry approach, we want to form a triazyl ring using copper 1 catalyzed cycloaddition. And we decided that it would be a good idea to form this triazyl ring just because it's more stable and also less likely to degrade. And so this is our new proposed synthesis using teremethine. Next slide.
So we'd like to thank everybody that was involved in this project. including the rest of our group members. We'd also like to thank Dr. Pazzi, Dr. Yamamoto, along with our advisor, Dr. Edward New, for all their help on this project. Thank you.